Yeah, sorry about that.
And we'd like to welcome everyone that's joining remotely. We're here at Whitehead. We'll be kicking off the CosMx presentation here for cell biology. And thank you for joining us via Zoom. Here in the room, we've got 20 plus participants looking forward to learning more about the CosMx Spatial Molecular Imager. And with that, I'm going to turn the time over to Maxine. This is Maxine McLean, who's joining us from the DC area today. And we've got a science-based presentation. We'd like you to be able to ask questions, specifically clarification questions on the content that's being provided at that moment. And at the end of the presentation, we'll open it up for just an open Q&A session. If you want to talk specifically about projects that you're working on, we can schedule some one-on-one time, perhaps, after the group has disbanded. And with that, turn it over to Maxine.
Thank you, Steve. So hi, everybody. I'm Maxine McLean. I'm a field application scientist. I actually I went to school in Atlanta and I started my FAS career over here at Emory. So I went to school at Tech, but I started as an FAS. This building actually was my first demo. So it's nice to come back and, just see some of the places I used to walk the halls at. And thank you all for having me. I am based up in the DC area now. And so it was glad to be back. And we have, as Steve mentioned, a deck about CosMx and about some case studies, some of the science that you're able to do with spatial molecular imaging. And as Laura mentioned, if there's clarification questions, if something isn't clear, please let me know. And if I'm looking at the slide, just say excuse me or get my attention. And then we'll have a Q&A afterwards for more discussion oriented. All right. So before I jump into the deck, let me go back. We just want to note the legal disclaimer. So this regards the ongoing litigation. I will say that I am a scientist. I don't really get into the legal matters. I understand that Emory Legal Counsel is in touch with nanoString Legal Counsel. So if there's any questions regarding the litigation, that it goes through those venues. And so on to CosMx. So CosMx is a spatial molecular imager, and you can see. Actually, does my mouse work? Yeah, here we go. So this is an example of the cells in the tissue that were imaged on this platform. And CosMx is part of the nanoString instrument suite, which is mostly genomics based. So nanoString is a genomics tools company based up in Seattle. We started out with the nCounter. I understand there's a couple of nCounters on campus here. It's a very simple gene expression platform. And then more recently, we have really focused on spatial. So it started a few years ago with genomics. It's a more tissue level spatial imager. There is a little bit of genomics data in the deck. And then CosMx is sort of the next iteration of spatial. So genomics was tissue level, select a region, you get some gene or protein content out of that area. CosMx gives you that gene or protein content within the cells, within the tissue. And both of those platforms have their data analysis in the Atomics cloud-based suite. Right. And so to speak about the general workflow, first day, you're going to have your tissue that's on a slide. You're going to, depending on the panel that you're using, you're going to incubate or hybridize that panel with your tissue. You're going to do some washes, general prep. And then day 2, you're going to load that slide onto the CosMx. And the CosMx is going to run. Basically, once you start it, it has the process for assessing the barcoding, and we'll get into some of the details, but it runs for a few days, up to about a week or more. And then that data is then uploaded to Atomics.
Researchers need to do anything once it starts running throughout the week.
No, it's really set it and go. And the slide prep is it can be over 2 days, but it's mostly one day that that is intensive for prep.
Is the gene hybridization done simultaneously with the immunofluorescence, or is it a stepwise process?
It's a stepwise process, yeah. Okay, and this is some example data, really just visually, where you can see the tissue, and the aqua lines are the boundaries that are drawn around the cells. And all of these other colors are, they're so numerous enough that they're blended together a little bit, but these are the individual transcripts that were identified in these cells. It's not all of the transcripts in this panel. Our panels are typically 1,000 genes, 6,000 genes. And so for imaging, you'll typically pick a handful of genes and you'll look at where they're distributed. And this is all available in our viewer. I actually have an example that goes through the process of somewhat data exploration. When you get your data back, how do what are the different visualization tools that are available to give you an idea of what are your cell types, what are the transcripts associated in that cell.
Really beautiful image. Say something about when cells are so densely packed together in a tissue. How do you distinguish between where one cell ends?
We have a section on it.
Very important. I'm glad you brought it up, but there's a couple of slides dedicated to it.
Oh, good.
And so to speak to the data a little bit, we have the UMAP plot here that looks at cell type and cell type genes. And then this is a corresponding plot that looks at the cell type map with the cell-cell interactions. The plot on the left is something that you would typically see for single cell data. It actually is single cell data, but it was generated off of CosMx and is part of the same data set of the graph that you see on the right. So you get the cell types, the clustering of genes, but then that data is then mapped onto the XY location where it was detected into the tissue. And so there's two outputs that you get when you're looking at the XY. You get the transcripts, which you can visualize, but then you also get the cell type that is generated through the cell typing algorithm from those groups of transcripts. And both of them tend to, depending on what genes you're looking at, the cell types tend to echo the morphology that you see in the tissue. The genes, depending on what you're looking at, do as well. But I have some more examples that will show some of this in more detail. And this just shows, that's a high level view. I believe the scale, several millimeters of tissue. And then you zoom into the multi-cell level. You can see individual transcripts here. And then you zoom into subcellular, and you can see where those transcripts are located within the cell. And so you can see subnuclear, outside of the nucleus. If you're looking at extracellular vesicles, you can see that they're outside of the cell, for example. But you get that very wide range of scale from tissue level to subcellular within these data. And we are compatible, CosMx is compatible with FFPE and fresh frozen. We have a couple of examples of tissue here. There's an imaging area. As long as the tissue fits within that imaging area, it's very common, for example, with mouse brain to fit more than one hemisphere section on the slide. We also have a tissue microarray. You can look at a number of different tissue types. It's really the footprint of the scannable area that determines the tissue that you're able to load. And these are just some examples that echo that previous slide. The images that we're showing here are the dots are the individual cell types, and you can see, as I mentioned, that they echo the morphology. If anybody's in neuro, for example, this mouse hemispherical section where you have some very characteristic markings, but you also have the variety of a tissue microarray, and then some of the diversity here on these different tissue types. So speaking of the scannable area, practically what can you load? That is the scannable area. It's a few, basically millimeter scale, tens of millimeters. And then you identify regions, fields of view to image within that. And we have some example runtimes. The CosMx has two formats. You can run 2 slides at a time or 4 slides at a time. And so you typically, all of our outputs are, for the slide set that you're running. But you'll see that the runtime depends on the amount of area that you're scanning and also on your panel size. So if you have a very focused panel, you know exactly what you're looking for. And we see this more with protein. We have a 100 protein ready-made panel. that's pretty quick. But when you're getting up into, you know, thousands, 6,000, we have a 6,000 gene panel, you know, that's going to be a longer runtime. More barcodes. Go ahead.
What area of flex are you with that 100 millimeter square?
Yeah. No, those are example. Those are not, yeah. Yes.
50, 50, 100, 200.
Yes.
Scan time very longer.
Oh, sorry. Yes. Here, I can repeat since I'm at the. Yes. Thank you for the reminder. Yes. So the question here was, is this scan area the only two options? And I would say that there are multiple options. We have a basically integral scan area. So you can, these are just two examples at either end of the spectrum. The question before that, somebody can remind me, or we're good? Okay, all right.
The RNA and protein analyzed on separate spots, or can you do it together on the same?
Okay, so here's my practice. Is the RNA and protein analyzed on the same slide or on separate slides? So for the panels, it's analyzed on separate slides. My understanding is that there is investigation into looking at it on the same slide. We have a combined protocol for genomics, but right now commercially it's separate.
Can you do where you could just look like these after the fact, like in general, you run together? If it's something like a serial section.
So we do have our analysis suite is either the protein track or the RNA track. There are a lot of third-party tools out there right now, and I will say Atomics is growing. Oh, actually, let me step back. Sorry, I'm already off track. Is that so? Just for the benefit of the Zoom audience, the question was, if you're doing protein and RNA together, are there integrated analysis software tools so that basically to combine those two data sets together? And the answer is in Atomics today, they're separate, but Atomics is expanding. And I would also say, you know, just looking at the amount of, you know, spatial analysis, you know, methods that are out there in the community, I would say that might also be a very interesting route to look at. Yes. 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 Yeah. So the question is, if you're looking at serial sections, how similar are those data sets? Yes. So you're right. There are, if you're running on serial sections and you're looking at matching up data from an individual cell, it needs to be from the same slide. So how thick of a section? Our recommendation is 5 microns. Yeah.
Us work on different disease models, and like for example, for neurologic disease, you might have a protein aggregate, or there's some sort of antibody that is a marker for pathology, and you want to ask the question of how gene expression changes those stuff. So for those types of questions, you really do need to look at the ability to multiplex. So that would be a future.
No, I completely agree. Yeah, no, and for the benefit of the Zoom audience, the point is that protein and RNA are intrinsically related, and so to be able to look at multi-analyte is key. And you know, when stuff is in R&D, we really can't speak. You know, it's in R&D, right? It's not a product yet, but we understand that that's of great interest to the market. Oh, yeah. So the question is the area / shape tunable? And so there's two ways to think about the area. So this area is basically it's like a microscope scan. It scans that area and your tissues on it. Within this, there is a square field of view that you select. And so you will select multiple squares, that comprise, you can tile it, or, that comprise your tool of view. So the minimum size for that is about 0.5 millimeter by 0.5 millimeter. And so it's tunable within, basically that's your unit of imaging. Okay, good. All right, great. I like the dialogue. Thank you. Okay, so these are some of our off-the-shelf panels. Before we talk about what is available to order, we also have full custom. So we do have, if what you're interested in doing isn't reflected in our off-the-shelf, one of the common strategies is either to look at the genes that are in the off-the-shelf offering, and then to add in the content that that you have that's not represented, or if you wanted to do full custom, that's also an option. The other thing is, I'm not going to focus on our protein today, but we do have human IO, a small human IO panel, and then we have a mouse neuroscience, which is actually used quite extensively. And there's a lot of good data out there. But just in the interest of focus, we're going to talk about RNA. Okay, and so before I get into the panel content, I just wanted to take just a minute and talk about the chemistry. So the way the detection is done is with in situ hybridization. Let me see if I can get the mouse over. Okay, well, you all will just have to maybe, there we go. Sorry, lost where this guy was. Okay, so this is your RNA that is in the tissue. And this blue line that's overlined with the red RNA in the tissue, that's your hybridization probe. It's pretty short. It's 35 to 50 nucleotides long. And then extending off of this, and so this is what is hybridized. That your panel would have designs for the RNA, complementary sequences here. You'll see that there's an alternating color sequence here in this tail that extends up. This is where the imaging happens. And so it's a series of, I'll refer to this as a flag. It looks something like a flag. There's this fluorescent flag-shaped tail coming off of it. But you'll notice that the stem is hybridized to the first branch on that tail. So we can do a series of different colors. And you can see by having basically a series of these flags, you can create a barcode effect. And so what we end up doing is we have a series of incubations, imaging, release of the probe, and then another incubation with the next, basically with the next station on that tail, to create a series of barcodes. And so there's 4 colors plus blank that enable us to have basically thousands of different individually barcoded nucleotide sequences. And then if you're doing protein, the only difference is the antibody rather than an probe. All right, and so our first panel is the 1K. So this is a very, I would say, it's become our general use panel. We use this in our trainings. All right, yeah. Thanks. Yeah. So it's 1,000 genes. It's for mouse or human, and it's great if you just want to get a first look at the tissue. It lets you do cell segmentation, but also covers some biology. And the content of the panel, you have cell state and function, cell typing, and then you have some additional genes for hormone activity, cell-cell interaction, and signaling pathways and target genes. So within that 1,000 plex, we really distributed it to address cell typing, which everybody wants to do, and also then do in-person biology with the cell types. So the question is, do we have this panel content online? I can't remember. We certainly make it available. Steve, I don't know if it's on the website yet, but yeah. We can share it with you if you're unable to just find it off of the product page. And so examples of the sorts of pieces of information that you can pull from the 1K panel. Cell atlasing, I would say, especially in the very early days of CosMx, this was the first thing that was the question with the new tissue type, is being able to type the cells. Neighborhood analysis builds off of cell atlasing. It looks at the cell types, but then which cells are proximal to a given cell, what are the patterns of those different cell associations. And then getting into, more standard biology, cell state and function. Going back a little bit to spatial, you can look at the ligand receptor interactome. And then conventional differential expression biomarker discovery. And then this is our same 1K, but if you're looking at mouse neuroscience, because atlasing is of such interest in neuro, and mouse is the primary model for that, we have a dedicated panel for this. And this is an example data set. I believe this is available on our website. So we have a number of data sets available. I'll just say in general, if you want to get your hands on some CosMx data, it's available for download. This looked at two mouse sections. So we have the coronal hemisphere and then coronal hippocampal cortical shown right there. And the example UMAP, as well as the number of the different cells that were identified typed off of the gene expression in the panel, the genes identified in the panel. You can see there's a number of excitatory neurons, a number of inhibitory neurons, as well as smaller, basically a wide diversity of cells in the mouse brain.
Single cell. The one question that I have is that because neurons are so highly polarized cells, glial cells, you have RNA that are glial processes and also neuronal processes in the neural. And then you also have nuclei that are not from the neurons, but from other supporting cells. I really whether signal is coming from the neuronal process or an adjacent glial cell that is embedded in neuron. How do you define the cell borders in the nervous system? It's so incredibly complex.
So the question was defining the borders in the nervous system and the risk of misassigning transcripts if that border is not correctly defined. So I have an example from neuro, actually a couple slides from here that will show you how some of the boundaries are drawn. I mean, I will say I haven't looked at the data myself, but when I think about the length of processes, most of the transcripts we're detecting are within some proximity to the nucleus.
Exactly.
Yeah.
Yeah. Software, that's a very good question. The software focusing on RNAs that are staying within so many microns of nucleus, and that's how it defines.
So I think the best way to resolve this question would be to pull up an image of the segmentation, because when I think about how long processes are, my initial, and actually for the Zoom crowd, forgive me, I'm sorry, that the question was really, like what exactly are the segmentation boundaries? Yeah. We have that data on the data set, so that might be a good thing to revisit. Yeah. Oh, so I will show you. Actually, maybe we can pick this up. I do have a neurosegmentation. Yes. I'm going to have to, Jasmine, are you on the line? So for the Zoom audience, what is the imaging resolution? I don't want to misanswer. I don't know if Jasmine or Nicole.
We have a few moments to look that question up and I can drop it in the chat.
All right. Thanks, Jasmine. Is it okay? Yeah. Well, and to go back to the the slide I had with sorry, I'm searching for the word, the way the fluorescence is detected. The spot size is, I believe, 50 nanometers. So, you know, yeah, some of it depends on the wavelength you're using and some of the other optics factors. Jasmine, who is one of our FASs, is basically my lifeline. So she's going to check it and then she's going to give us the answer. Thank you so much, Jasmine. But yeah, that we'll have, and then we'll come back to this when we're on that side about that.
I've seen it referenced a couple of data sets in the presentation. I'll be sending both of those out. We liver data set and a mouse neuro.
Thank you, Steve.
Take up the entire afternoon with
This is just a quick note on cell typing. We do have 3 different modes. You can go from supervised, semi-supervised to unsupervised. It just depends on how much prior knowledge you have on that data set. And we do have, we did publish our methods it's in bioRxiv right now, but that's available if you want to dig into it a little bit more deeply. Segmentation, we do have a section on it that will dive into segmentation in a little bit more depth. But since we were talking about cell typing. Okay, so on to 6K. So a little over 6,000 targets. You can add with 1K, you were able to add 50 targets. Now we've increased that number to 200, so it gives you a little bit more bandwidth. If your genes of interest aren't already in that 6K total. And the way 6K was designed, so that this is a human panel, but it was designed to cover nearly the entire reactome. And this is a map of the reactome. The darker coverage is basically the higher the coverage is, the darker the green. But the reactome is the map of all of the distributed pathways in human biology. It's available. It's a public resource where you can go and basically look up the different pathways. And so we have covered it to really we designed the panel to maximize coverage of the genes that would be associated in cell processes. And so this is another data set. So this is human cortical tissue. Again, I guess, Steve, if you're sending out data sets, this would be a good one to add to the list. But you can see that this is just a preview. So not only do we have the panel list, but we have data that you can interact with off of these different panels. And this is some example data off of 6K. And this was basically the nanoString R&D team stepped through I think of this as sort of the, when you're first looking at a tissue, how do you assess it? What are the questions you ask? And so that's what this set of slides goes through. I believe this was a section from melanoma. And just a quick take, this is that middle section that has the sort of the rainbow of colors. That's the whole tissue. And then we have a few zoomed in views. We have the top one was cell states. There were a number of cancel, basically when we were doing the cell typing, there were multiple states of cancer cells that were identified from the differences in gene expression. And then when you look at the other region here, this one that's mostly blue and red, you can see a combination of tumor and what I will assume is stroma, and you can look at the tumor-associated macrophage response. So with the single cell resolution in the whole tissue, there is a wide variety of different things that you can ask of that tissue, and this is something of an example walkthrough. Okay, so here's that overall tissue. You can see the transcript density is relatively uniform. There is some variation as you might expect, but basically we image the whole tissue here. This is a quick list of the cell types that were characterized off of the 6K RNA data set. And then one of the things that when you're first looking at these data sets, one of the questions is, how does the RNA and the cell typing match up to the immunofluorescence that I use to identify my cell boundaries? And so what we have here at the top is a series of the morphology stains that were used. And so in this experiment, there's CD45 and cytokeratin and CD68. And we can see here, so these are some characteristic genes that were identified for each of these different regions of the tumor. So, broadly immune, cancer, and then macrophage specific. And then you have the cell types that resulted out of that. And so you have, this is a vein of plasma cells here shown in blue. And when you're looking at this in the software, oftentimes you will toggle back and forth between the cell state and then the genes that you see overlaying on that same view. And then we have 3 different types of keratinocytes that were identified, and you can see this diversity in genes that were identified in the same region of the tissue. And then again, multiple different subtypes of macrophage in the overlay of. I apologize, this color contrast isn't the best for the macrophage, but you can see that there are multiple macrophage subtypes identified from the in situ site. And then this was the other region that the team looked at. So this was called a keratinous kill. So this is an abnormal squamous cell organization that is found in some skin cancers. And then you have this overlay. These are the different cancer cell types that were identified from the in situ typing. And you can see that there is some organization here, and the poster doesn't really go into it, because this is really a demonstration of the technology. You can see that there is some organization. Some of these cell types are found more radially. Some of them are found more closely around the keratinous pill. And then you also see the overlay of the genes. And so we have a short list of genes that are shown here. One of the fun things to do is normally you have, at least in your head from the literature, some key genes you want to look at. You pull up those genes, you run differential expression, you pull up the genes that are differentially expressed. And so it's there is a fun to look through the data and see how these different genes are expressed throughout the different cell types and throughout the tissue. All right, and then one of the tools that we have in Atomics is the in situ core analysis. And what in situ core does is it looks for genes that are correlated spatially in the expression, use of some sort of co-localization. And so what we see here with this highlight is that if you go from the gene analysis to the cell type, you see this interaction between macrophage and keratinocytes. And then this is the analysis of macrophage within the tissue. So the cell types, we have cancer cells in blue, macrophage in red. You can see most of the abundance of macrophage in this center region here. But then there's a few in the tissue. And if you're just doing a reality checking thing, are these macrophage expressing what I expected to express? You look at genes associated with tumor-associated macrophage. These are some that are just available from the literature. And you can see for the yellow is the one that shows up best, but for example, the yellow is abundantly expressed in these regions where you have a macrophage cell type within the tumor. All right, and then, so that was a demo of the 6K. This is a case study that uses CosMx to look at simian immunodeficiency virus as an analog to HIV research in non-human primate. And this was out of, this was out of Jake Estes' lab at OHSU. Okay, and so what was the motivation? What's the general context? So this is an analog for HIV, and you have treatment and control. So and then infected and non-infected rhesus macaque. And the treatment was an anti-CD21. So the virions bind to CD21 as part of their infection model, and there's some evidence that we can outcompete that engagement with anti-CD21 therapy. And the idea is that would then eventually reduce or remove the viral reservoirs. And so the question was spatially, what is the biology at these regions of viral infection? So if we can spatially identify where the variants are in the lymph nodes, and how does that change with treatment? So So, you know what? So I was remiss when I was talking about protein. So I wanted, and so the question for the Zoom audience is, how do you look at, if you can't look at protein on the same slide, then how do you identify where the virus is? So you can look at protein in the same slide. So there's two ways to talk about protein. There's the protein panels, which are relatively high plex, especially for protein. And then there are the morphology markers, which is the immunofluorescence staining. And so those are, 3 markers. Yeah, so you can do some protein. You can also do RNA, so actually this was a viral, if we look at this one actually, the staining that they did to assess the morphology of the tissue. So there's a nuclear stain, CD20 for B cells, and then the viral particles was actually an RNA probe. But you could do a protein if you wanted. And that's exactly what they did, was they identified where the virions were, and then they used that to direct the spatial experiment. Thank you. Okay, so shall I repeat that? So that, I guess for the benefit of the recording. Okay. So let's see. All right. So to talk about the preliminary data that was done on geomics. So I mentioned geomics is more tissue level. They picked regions where they had identified basically virion presence with the staining versus areas that were negative for viral presence. And this was done, I believe, in the terminal centers of lymph nodes, but yeah, maybe we can double check that in a later slide. But anyway, what they did was they did this in treated and untreated monkeys, or treated and untreated macaques. So they had infected and uninfected, and then they also had within the infected, treated and untreated. So within the treated infected, there were still positive and negative regions. So there's a couple of different subgroups here. But if you just look at the total clustering of variant positive versus variant negative, you can see that there's still a differential response, with IL-10 being one of the significant differences. And if you do a pathway analysis, you find that IL-10 is actually a modulator for a number of the other different differentially expressed genes that are found in this biology. So this is not single cell. This is basically, it takes that tissue and it says, what are all the genes associated in this region? The next natural question is, what are the cell types, or are the transcripts associated with different cells? And so that's when CosMx was brought into play. And so you can see here's the scan of the tissue. Those yellow boxes are the fields of view that are selected. This also gives you an idea of how you might go about doing an experiment. You can tile. What I have seen in practice is that normally there is especially if we're dealing with tissue that has an architecture, there's a region that you want to assess, and you make sure over your samples that you're capturing as similar region as possible over those areas. This is we have the fluorescence here. So we have, I believe these are B cell and T cell markers, as well as the marker for the viral RNA. And so the CosMx was stained for these immunological and viral markers. The process was run. This is a preliminary UMAP projection that came out of that data, as well as some initial cell types. And this is a closer view. And what you can see, it's a little, you have to, with the number of points here, you have to look a little close, but you can see these red dots indicate actively infected cells, positively infected cells. And so this is an untreated control where you have basically a high level, at least in this local region, of viral RNA. And then in the treated sample, similarly, you see a relatively lower than this. You can still find some red dots, but overall the viral RNA is lower. And then when you take this into the analysis and look at it further, so looking at the B cell follicles and then the T cell zone, so there are two different regions here on these two graphs. And you can see that the way we've grouped the differential expression, we're comparing infected cells versus uninfected cells. So the segmentation, there's the tissue, there is the cell assignment, and then the cells are grouped. And if the cells are infected, and you can see some examples over here on the right, where you have the blue cell boundaries and then the bright red dots indicate positive infection. So the transcripts that are associated with those cells are in one group, and then the uninfected cells in another group. And you can see that, so we're looking at, we have 91 cells and 134 cells in the infected groups. And then basically there's a very clear differential expression in both of those groups. And then if we go to how the tissue is organized, in this movie, we may have to play it a couple of times, but there's a couple of views that you have here. So it switches between the genes and the cell type and what's called the TME niche. So that's the cluster of cells that it generates from the analysis. And those three types of data are then overlaid in a UMAP as well as within the tissue. And so you have a wide diversity of data visualizations that are available with single cell data in the Atomic suite. Yeah, I will say, there's something about, when you take it and you put it up on a slide, it's not as interactive as it is when you're actually looking at your data and you can toggle back and forth and you can zoom in. It is, you go from that very high level view. I think I've said it before. It's just an experience.
This is coming from our forum. How much of that is on us?
Yeah, so for Atomics, so the question is, how much of this is plug and play and how much of this is, here's your raw data, have at it.
And
Yeah. So we give you both. And I may have to defer to Jasmine. If Jasmine, please fill in if I am missing information here. But what Atomics has right now is a drag and drop analysis pipeline. So we have certain modules that are already built into the analysis, and the data is already in Atomics. And so that lets you run cell typing, and I believe there's ligand receptor analysis. We have a few more modules that are in development now. And then we also have a data download option with the Seurat object. And so we make both options fully available. And I will say, it's been my experience. I did work with single cell a few years back, and there's the tools that the community is making are always more advanced and more specialized. And so I think that's where a lot of the interest is going. And I expect in the next few years that we will have more of a spatial imaging community that is focused on providing those tools. But as those tools, have demonstrated utility, they will make their way into atomics, is what I expect will be the path. But that's kind of it's the originality and creativity that you see in with primary users is what's going to drive the innovation. Aside from this, we're going to have basic suite.
The comments is going to allow for basic analysis, and we've seen a massive growth in the past year of companies that are popping up, specialized specifically on system analysis. So we're never going to be able to keep up with like the niche company that's what they focus on 100% of the time. However, Atomics is very robust for what it is, and is able to provide onboard analysis in the cloud with Atomics.
So the atomics is for considering a core. Is the atomics something that is part of the core that is everyone has access to, or is it going to be specific that's how fast?
Good question. It's, you guys get to administrate, who uses and who sees what. But it is an AWS cloud-based repository. You guys have full control over who you grant access to. Certain labs can have access to only their data. Other labs have it.
Is there a short data when it disappears after a certain period of time? What are the charges for storing the data in Atomics?
So the data does not disappear in Atomics. It is up there. I do believe are options for cold storage, if you want to archive things. But yeah, and we can get you more information if you're interested in the specifics. But yeah, it is AWS. There are charges if you have a lot of data. It is something to take into account.
I'm sorry,
Oh.
What's the way that this actually is?
Okay, so to take a step back, the question was, what are the limitations of the slide prep? So we have protocols for FFPE and fresh frozen. So typically fresh frozen comes from an OTC embedding. That's not a problem. There is a fixation that helps adhere the tissue onto the slide after you have done some preliminary washes. For FFPE, the first step is to remove the paraffin. I mean, it's a pretty conventional procedure in that sense. And then we need to go back and look at the protocol, but I would there is an antigen retrieval, just to enhance the antibody activity. Mm-hmm.
So you got your have access to it already.
I think most people that are doing
Okay. You're not going to run into anything that's complicated with this one. You can run IC, you can run CosMx.
And we have a few minutes left, so let's take a look at segmentation. So this is something that the R&D team has spent a lot of time working on. I'll say, just off the bat, segmentation, it's never going to be 100% perfect. And we always recommend when you have your data set to have a strategy for verifying the accuracy of your segmentation. And Steve, if you're going to send out the article on, just using your spatial imager, it covers, we have guidance for it.
A very important topic. I know we're kind of up against the clock, but cell segmentation, when you look at nanoString versus 10x and how we approach cell segmentation differently with companies, that's a big part of different cell segmentation.
Yeah. And some of the core algorithms that we use in our segmentation, so we have CellPose for RNA and Celesta for protein. These are both developed in the community and available in publication form. We have integrated them into the Atomics workflow. So the segmentation, this slide actually speaks to it. So starting off, your tissue has a nuclear stain. It also has a morphological stain, and I'll have a couple of examples. But so you have your nuclear model, and then you have, cytoplasmic model, which surrounds the nuclear. And this is fed into an algorithm, which has been trained on, we have several different tissue types that we have. There's different, there's a neuro option, for example, but there's different tissue types that we've trained this model on so that we have specialized segmentation strategies. And the segmentation is also based off of a Z-stack image that's acquired as part of the imaging to improve the overall segmentation quality. And so when you're selecting your segmentation, you select the trained algorithm version that you want to use. It generates this final output. You can see these lines here around these cells. And you can see it's captured a few different shapes here. These are some, all of these samples are somewhat similar. The cells are closely packed. What I did want to point out is the different staining options that are used, in this pretty generic segmentation. So you start out with a nuclear stain, which we refer to as DAPI, and then this mouse brain was ribosomal RNA and GFAP for astrocytes, and then for human, CD298. So this is it's associated with the potassium sodium pump. So it's broadly expressed, in general human cells on the membrane. This is a pretty sort of all-encompassing marker. And then we have pancytokeratin as well. And so you can see these are examples of segmentation on these different cell types. This is a neuro example. So this is using our conventional markers of ribosomal RNA and GFAP. You have the fluorescent image. You have the fluorescent image over on the far left. This is the segmentation. And then after cell typing, basically that cell type is overlaid within the segmentation boundary. And so you can see, with the simpler processes, that it is able to draw those outlines very efficiently. I think where the question comes in, I think this is where the question earlier was alluding to, was if you have these very extended processes, is it able to, trace those? And right now it's, we can double check the data, but my understanding is that that's not the focus of this. It's for body, cytoplasm that is relatively close to the nucleus. This is the more advanced segmentation. So using additional markers to help draw the boundary that is not available just with the general GFAP ribosomal RNA. And you can see here that it's a combination of different cell types, but that these markers are able to follow these processes relatively accurately.
So one slide where I would want to be embedded in your mind. This slide right here. And now we can truly identify the single cell. No, I'm like, why don't we step out? This was the thing.
And so Steve was mentioning this earlier. In comparison to 10x, this is where we put a lot of our resources on instrument performance, is getting a very accurate cell segmentation algorithm. And so comparing it with polygonal expansion, you can see here that, the polygonal expansion is, it approximates the cell boundary. Does it do the fine processes tracing? No. And so that's the difference in CosMx cell segmentation. There's also some additional factors that I'll point out, but and I think that everybody's aware, just because this has already been brought up in the group, but if you have this general sort of expansion boundary setting, you can get misclassification of genes from the edges of other cells, and this will dilute the accuracy of your cell typing. And so I'll go back and say again, no segmentation is 100% accurate. There's always going to be some, just guessing, some variability in the system. But this is also something that needs to be you can quantify it. It's not a mystery. You should have a plan in place for quantifying it.
Image files or what is the data?
So there's a couple of data formats. There are image files. Right now, so the question was, what is the data format that the the data is in? So the data, the counts are in a direct object, as well as I believe some of the metadata like cell typing, but I could be mistaken there. And then the images are also available as well. There is a Z stack that you can export. It is not yeah, just thinking about the Z stack. So right now that's something where you can get specific instructions for downloading it. Now, what I will caution is if you wanted to run your own segmentation algorithm, CosMx right now does not say, okay, use your added segmentation algorithm. There's the algorithms that are already validated. And so you're choosing from one of those. If that was where I don't know if that was where the question was going.
Yeah.
Yeah. The question is, how do you come up with you're saying,
Well, and I would also, yes, I would say in general for segmentation, it is part of CosMx. If you're going to build your own pipeline, we give you the counts with the coordinates. I would just, my understanding is that it's non-trivial to go from that unprocessed data again. But to look at the differences that segmentation quality has on your data, this is a comparison of CosMx versus Xenium. This was done from a researcher, Dr. Suzuki at University of Tokyo. You can actually look at this data set on the link that we provided. And he ran a comparison data set of CosMx versus Xenium. And just some of the initial QC, found that, in general, the size distribution on CosMx is a much tighter distribution. And is also tends to be, if you do the bout calculation, this is the total area. It tends to be more representative of the cell diameters that you would expect conventionally in tissue. And we can see that. I mean, even in these boundaries, you can see that they're a little larger than probably the cell is. And then if you look at some of the data. Okay, here we go. So this is more of a plot of those size values. So CosMx, you see that the distribution is relatively tight. There is some spread, but they're more outlier spreads. And then on Xenium, you do have some of the smaller cells as well, but then you have a wide tail, basically of larger cell sizes. And one of the cell types that has the larger tail is lymphoid. And from what we expect, we don't expect the cancer cells to be the small cells and the lymphoid to be the large cells. It's actually the reverse. But we don't see that in the Xenium data. And so what impact does this have on the data quality? Well, this is just a simple, autocorrelation map of these cell types. And you can see, you compare CosMx cell types to Xenium. The data is not, there is a lot of similarity, for sure. But if you look at the correlations between these cell types, CosMx tends to have more localized correlation along this axis. These cell types that are not close to each other, the blue is the anti-correlated or uncorrelated, and you can see that there's large swaths of blue. There is less of that in Xenium. You also, it's a little hard to see here, and I'll share these slides later. You can look at it on the website, but if you look at the ability to differentiate CD4 versus CD8 cells, for example, cells that are similar but are also very different, there is basically way more overlap in the Xenium data set than there is in the CosMx. So these are just a few snapshots of the data that's available on this website. Again, this is from a researcher at the University of Tokyo who ran a comparison. I know there's a number of comparison studies out there. Yeah, I would say, if you're selecting spatial systems, I think it's important to be aware of the performance characteristics of the platform that you're using. And with that, I think we have a few minutes left for questions. we're a little bit over time. Thank you, everybody, for your attention. Yeah.
These 6,000 genes, are they the most abundant cells of what of these 6,000?
So we picked it to cover the reactome. I think it was to cover, there's about 20,000 genes in the whole transcriptome. We covered it to bias towards the genes that are associated with cellular pathways and processes.
Speaking of the whole transcriptome, you guys will like to see on LinkedIn presentations next week. We are releasing CosMx full transcriptome data at AGBT. That can be commercially available for sale until 2025. However, R&D has been working on that for several years. And I think they just cannot wait to let the world know that we have achieved full transcriptome sequencing. That's going to be the budge for us at AGBT, but 6,000 flags is still, way above what anyone else can do, and it's commercially available immediately after AGBT.
Yeah, that's a good reminder on whole transcriptome. It's coming soon.
You got to give the R&D guys their, their spot.
So that being said, do you have an idea of how many like the genes we can detect using the single cell RNA-seq?
So the question was, how many genes can we detect with single cell RNA-seq?
Yeah. What's the sensitivity?
So I don't know of a graph that looks at that offhand. I know that there are graphs that look at they do have single cell data and CosMx data. So actually, no, now that I mention it, so the question is, yeah, how do you compare single cell data to CosMx? There are comparisons out there. I don't remember what the sensitivity is, but yeah, if you have single cell data, you can apply that to your tissue and make the comparison. But they're out there. I would say that information is out there. We just, yeah.
Yeah, can I jump in and just say something real quick? So there will also be a seminar on the 25th this month. It's going to be over in Dementia to talk about the tenant data. So I would really appreciate it, if you want to attend it remotely, to please, because I like your input from, I have to do my due diligence for both systems. So please consider attending that one as well, so that you have both sides and you can see how we object. Take a look.
Right, if you want to speak to the Zoom audience of that saying, because there's way more people on Zoom. Oh, you did. Okay.
Yeah, and I figured we were, I don't want to interrupt the questions, but I do want to catch the folks who are leaving to remind you that there's another seminar that is on the 25th. It also has, like I said, it's going to be hybrid, so you don't have to walk all the way if you don't like it, but yeah, so, please give that one some time as well. Thank you for coming. Something go back to the question. I was just want to catch.
And I appreciate everybody for staying over time. Yeah. Thanks for your consideration.
Yeah, it's going to be shorter.
Great.
Yeah, we have.
Stop recording.